Okay, we're back. Let's see where I was. Okay, so in this project, we wanted to investigate the prospective phenotypes reachable by individual amino acid substitutions on the same gene and see how differently can they impact underlying cellular programs and states. So we decided to focus on the RUNX1 protein, which is a transcription factor, part of the heterodimeric transcription factor complex core binding factor, which consists of the, oh, actually, let me have the pointer. Sorry. Perfect. Which consists of the DNA binding RUNX1 and non-DNA binding CBFP proteins, and heterodimerization of CBFP to RUNX1 increases its DNA binding affinity. And RUNX1 is a master regulator of hematopoiesis, and as such, it regulates cell proliferation and differentiation. So it's a very important gene in regulating cellular identity. And the importance of the most of these functions happen through the RUNX domain of RUNX1. Through this domain, RUNX1 binds to many promoters and enhancers, binds to CBFP, and also interacts with various other transcription factors and co-regulators. And the importance of this domain is reflected by how frequently it has been targeted in cancer. We can see various hotspot mutations on the domain here. And due to its importance in regulating cellular identity, RUNX1 has been frequently implicated in leukemias, but also has been shown to contribute to breast cancer. So in this project, we wanted to understand the potential of different mutations on RUNX1 to generate distinct functional effects in terms of how they modify different cellular programs and states. And Perturb-seq provides a tool to investigate such perturbation effects. Ranging from whole gene knockout to single amino acid substitutions by studying the transcriptomic space. So we decided to use SEOs, which is a Perturb-seq style approach developed by Dr. Prashant Mali's lab at UCSD, which uses a bar-coded open reading frame overexpression libraries coupled with single-cell RNA sequencing to assay cell state and fitness. And the key point here is that each library element contains a unique barcode sequence, so you can map each cell to a single element after sequencing. So these tools are powerful and scalable, but they're still expensive for exhaustive mutagenesis, meaning that we cannot just generate every single amino acid substitution on RUNX1 and profile them. We still need to carefully select a subset of mutations to study. So we decided to perturb distinct interactions of RUNX1. So for this purpose, we created a structurally resolved interface map of the RUNX domain of RUNX1, where most of the interactions happens. And in this map, each row represents the 2D amino acid sequence of the domain, where interface residues that are involved in the physical interaction with each protein partner of RUNX1 is highlighted black. We were able to structurally resolve 33 interactions of RUNX1 that had available structural data. And using this web, we created the RUNX1 subnetwork with RUNX1 in the middle, all the partners on the edges, and the interface residues of RUNX1 that are involved in each specific interaction are triangles here. So we identified 83 residues within the RUNX domain that mediate at least one interaction of RUNX1. So we created an open reading frame library to perturb each residue. So in this experiment, it was designed to overexpress a vector containing the RUNX1 variant with a barcode sequence unique to each variant. And we used a chronic myeloid leukemia cell line where RUNX1 is the regulator of cell identity. And we knocked out the endogenous RUNX1 so that we can see the effect of RUNX1 mutations more clearly. The library contains a total of 117 elements with the wild type and loss of function control constructs, the 83 perturbation variants I mentioned, which is our main interest, various combinations of them, and additional negative and positive controls. So all of these mutations were sequenced in the single cell setting, and then for a select number of mutations, bulk RNA and ATAC-seq experiments were performed to validate and for further study of the variant impact on the RUNX1 regulome. But I will only talk about the single cell part today for time constraints. So we hypothesized that variants with different effects on RUNX1 targeting should cluster separately in the transcriptomic space, and we have the wild type and loss of function control constructs to provide a frame of reference for mutational impact, for functional impact. So we After quality control filtering, we recovered around 20,000 cells with detectable barcodes assigned to a single cell only, single variant only, sorry. So each cell contains only one RUNX1 mutant. And we have a median of 136 cells per variant with a minimum of 22, which has been shown to be enough to detect variant effects previously. Interestingly, we see that the loss of function and positive control variants generate higher number of cells than wild type and the negative controls, which actually is consistent with previous results that RUNX1, acts as a tumor suppressor. So loss of RUNX1 will activate cell proliferation. So here we can see all these single cells where UMAP provides a two-dimensional projection of the gene expression. And the unsupervised clustering of single-cell gene expression profiles reveal 3 clusters. Cluster 1 contains the cells with wild type and negative controls, while clusters 2 and 3 were enriched in cells with loss of function and positive controls. Perturbation variants, which is our main interest, seem to show bias towards different regions of the UMAP, but it is really difficult to study all of them all together. So we decided to pseudo-bulk them and look at differences of mean expression profiles of each variant. So here, we, instead of looking at the single cells in the UMAP, we are looking at each variant separately. Each dot represents a variant. And again, unsupervised clustering of these variants reveal 3 clusters, with cluster 1 containing the wild type construct in all of the negative controls, and cluster 3 containing the loss of function construct and the positive controls. And then the perturbation variants, which are highlighted in gray here, also seem to fall into these groups, but separate assignment of 14 of them in this middle group suggested a partial loss of function and possibly a gain of new function, different from a complete wild type or loss of function transcriptional state. So we label these groups accordingly as wild type like, hypomorphic, and a loss of function like. And revisiting the single cell visualization with these labels, we were able to see that these 3 phenotypes, different phenotypes, occupy different regions on the single cell setting as well. To quantify transcriptional effects of mutations, we decided to use Hotelling's 2-sample t-squares test, which is a student's t-test generalized for multivariate testing. We compared expression profile of each variant against the wild type or loss of function control construct, and used the T2 test statistic as a distance metric, where higher scores indicate a higher deviation from the control being compared. And using this statistic, we saw that all of the wild type-like variants were indistinguishable from the wild type control itself, with very small T2 scores against the wild type and high T2 scores against the loss of function control. And this opposite was true for the loss of function-like variants. But the hypomorphic variants, on the other hand, were significantly different from both controls, which suggested a not just an intermediate between wild type and loss of function controls possibly, which was supported by differential expression analysis, where we saw various genes being differentially expressed in the hypomorphic group, but not between loss of function and wild type controls. Fitness analysis also showed that loss of function groups had higher fitness than wild type-like group, which is again consistent with RUNX1 acting as a tumor suppressor and activating cell proliferation. Okay. Next, we performed hierarchical clustering of genes across each variant using mean expression profiles of variants. And the resulting dendrogram revealed the transcriptional differences between the wild type, hypomorphic versus loss of function-like groups. To better understand gene expression programs distinguishing these 3 groups, we looked at functional enrichment of genes associated with each. Group 4 was upregulated in wild type, gene program 4 was upregulated in wild type-like group and was enriched in immune system-related functions, which is consistent with RUNX1's role in hematopoietic lineage commitment and differentiation. Programs 1, 2, and 3 were upregulated in loss of function-like group, and they were enriched in heme biosynthesis, NGN, vasculogenesis, and ECM regulation terms, which is consistent with loss of RUNX1 leading to erythroid differentiation and hematological malignancies. Hypomorphic groups, on the other hand, seem to be upregulated in program 9, which was enriched in tau protein kinase activity, which has been suggested to link NGF to activation of MAPK signaling. But the group was downregulated, in the gene program 7 was downregulated for the hypomorphic group, which seemed to be enriched in neuronal plasticity and exocytosis terms, which suggest variant effects on some of RUNX1's actual, less well-known functions for the hypomorphic group. Next, we use an NMF non-negative matrix factorization approach to infer transcriptional patterns that are active in each cell. NMF aims to infer groups of co-regulated genes across cells by factorizing the gene expression matrix into a gene by pattern and pattern by cell matrix. And by looking at the cell matrix, we can study cell membership of each pattern. And then by studying gene membership of each pattern, we can look at which pathways are activated in each. So we use CoGAPS and NMF-based technique and identified 7 expression patterns. And we can see the correlation of each one with our previously inferred phenotypes here. And then by using the pattern by cell matrix, we looked at which patterns are active in wild-type-like versus hypomorphic versus loss-of-function-like groups. So a quick look at each of them. Pattern 4 was upregulated in wild-type-like group, which you can see from the box plot here, was enriched in immunolatent functions. And pattern 3, most active in the loss-of-function-like group, was enriched in heme biosynthesis functions consistent with our previous results. But for the hypomorphic groups, interestingly, patterns 2 and 5 were most active in these groups. And pattern 2 was enriched in G1S cell cycle and senescence terms, which could be indicative of a G1 cell cycle arrest potentially for some of the cells in this group. And pattern 5 was enriched in interleukin and cytokine signaling, which could suggest a shift towards a more inflammatory state for some of these cells. Next, we performed 2D location-based phenotypic profiling of all of these variants. You can see it in the 2D amino acid sequence of the runt domain here. And on the y-axis, we have the T2 scores to provide a phenotypic impact against the wild type control. And we saw some clustering of functional variants highlighted in purple here, which was reflected in the 3D structure as well. So based on this result, we decided to investigate the impact of mutations on the DNA and CBFB binding regions of RUNX1. So we identified 11 DNA binding residues of RUNX1 by using the distance to nucleotides in the 3D structural space as within 3.5 angstrom distance, and which are highlighted red here. And we identified 19 CBFB binding residues, highlighted magenta here, as contributing to the binding energetics at this interaction. And then we saw that DNA binding residues were highly enriched, significantly enriched in functional impact, suggesting that interruption of DNA binding is highly damaging to RUNX1 function, as can be guessed. Binding of interruption of CBFB binding was also damaging, but not as severe, which reflects CBFB's role in terms of increasing DNA binding affinity of RUNX1. Next, we evaluated RUNX1 mutational landscape in cancer. We collected mutations from the COSMIC database and saw that even though mutation frequency was not highly correlated with the expression-based phenotypic profiling we performed, cancer mutations were highly enriched for functional impact, and the higher the frequency, the higher the impact. And actually looking at the 5 most frequently mutated substitutions here, we can see 4 of them are loss-of-function-like mutations that are involved in DNA binding, and 1 is a hypomorphic mutation that is involved in CBFB binding, correlating with our previous results. And next, we looked at the primary tissue landscape of these mutations and saw that majority of them were involved in hematopoietic and lymphoid malignancies, followed by breast cancer, which is in line with RUNX1 role in AML and breast cancer. Okay. In conclusion, in this project, we looked at the effects of distinct protein interface perturbations in cancer using single amino acid substitutions and analyzing the transcriptomic space at the single cell level. We showed that we can identify functionality of the variants based on single cell expression profiles. We also showed that functional RUNX1 variants target DNA binding very significantly. They're recurrent in cancer, and they're associated with higher fitness scores, suggesting RUNX1 acts as a tumor suppressor in K562 cells. We also interestingly found a potential gain of new function in a subset of the variants, which we labeled as hypomorphic. And they seem to implicate some of RUNX1's less well-known functions, such as NGF-stimulated transcription, neuronal plasticity and exocytosis, interleukin and cytokine signaling. So overall, this project demonstrates the potential of targeting protein interactions to define the space of cellular phenotypes reachable by single amino acid substitutions. But it is also important to note here that in the case of RUNX1, we saw somewhat subtle effects. We were only able to identify 3 distinct phenotypes, which could be due to evolutionary constraints on RUNX1. But another gene using the same analysis can potentially provide a higher range of transcriptional states reachable by point mutations. And with that, I want to take my advisor, Dr. Hannah Carter, along with my collaborators, Prashant Mali, Rebecca Panvala, and Gina Sheen, my lab, and I want to thank you all for listening.
Many thanks.
I don't know if I have time for questions, but...
Let's take one question. And also, Lisanna, if you don't mind setting up the next speaker, this may be safer. So, Kivilcim, any question to Kivilcim? Yes.
Great talk. And in the beginning, you had this illustration suggesting that different parts of this cellular network would be affected by different mutations.
Yes.
Did you look into this, whether you can actually quantify this, whether these different transcriptional states also correspond to different areas in this network?
Oh, yes. There was some correlation, but not, oh, sorry. There was some correlation, but because we use predictions to create that structurally like interface map, it wasn't as strong correlation as we hoped in the beginning. That's why I didn't show results. But yes, there is some correlation, especially for a few of the important partners of RUNX1, which we will include in the paper, which is soon to be online, hopefully. Thank you for that question.